Hi, everyone. So we're going to give people a couple minutes just to be able to get into the breakout rooms. I know it takes a little while sometimes. But in the meantime, while you're waiting, you're all welcome to go ahead in your browser, type in umgear.org. Joshua just kindly put that link in the chat box because during the presentation, I'll be going through some slides and then you guys can also follow along on the actual website. So we'll give it about 1 or 2 more minutes. We'll see if people are still joining, then we can go ahead and get started.
The analysis 101.
Pardon?
Not the, I want to break out room 2. Eric,
In 2? Okay, so go ahead and click F, you see at the bottom of your screen and leave breakout room. It will take you back to the main room and then you can click number 2. Or if you're not able to click, I know the help desk should be able to assign you into it. Yeah. Okay, it looks like people have stopped coming in for the most part. Maybe there's a couple more. I'll go ahead and get started. If anyone's still coming in, we have slides that you can follow along with. So I'll first go off. So hi, my name is Gwilliam. I'm a 5th year PhD student in Rona Hartzano's lab. And then this is Joshua Orvis with us here. He is the main brains behind the gear and making sure everything is always functioning properly. So the way this session is going to be run, it can be very interactive. So if you have a question at any point while we're working through this, just stop me or message in the chat. Joshua is going to be looking at the chat if there's any questions, and we can go through any question you may have. So it is very informal in the fact you don't have to just sit and listen to me talk the whole time. We want any question you have to be answered as we go along. So for anyone who's new that may have just joined, if you haven't already, go ahead and open up the umgear browser, so umgear.org. It's in the chat box, the link there. And then I'll go ahead and start going through these slides. I'm not going to have them full screen just because it's a little bit easier to toggle back and forth between gear with them. Okay, so first things first, welcome to gear for anyone who hasn't used this before. If you have not used gear before or do not have gear, we highly recommend creating an account. This makes it so that you can really customize the gear portal yourself to different displays, upload your own data sets, and have everything saved for the next time you go on. So why create an account? Really it makes it a lot easier to use the analysis workbench. There's data set note-taking, generation of gene carts, uploading of your own data sets, and creation of your own custom displays of existing data sets. Some of this we'll cover in this session, others are in other sessions. So after this breakout room, if you decide you want to go into a little bit deeper, you can join one of the other rooms in the next session. So in order to register an account, you want to go on to the top of the browser and click the little Create an Account tab in that right corner. And then it's just going to come up with a form with basic information, first, last name, institution, and then creating a password. We don't share this information with anybody, so it's safe and secure. We're not going to give it, you're not going to get phone call spams from this. And then from that, you're just going to click the create account button. If you ever do forget your password and you type in your password, there's an issue there. Then you'll see once you type it in, the little sign in will come up with an error, and then you'll be able to reset your password from there. And so then once you sign in, you'll automatically be signed in. Some computer browsers will constantly remember your password, others won't. So that just depends on your computer. And one thing to mention that Rona mentioned in her presentation is gEAR works best with Google Chrome. So right now, if you're using a different internet browser, I recommend switching over to Google Chrome. The displays and everything have been designed to work best with that. And if I'm going too fast for anyone at any point, stop me and we can pause for a second to make sure everyone's on path. So the real reason the gEAR first was designed was to be able to easily explore gene expression among different data sets in one convenient location. So you can see when you go on the home page here, the very first thing that's on this page is a little search bar for a gene symbol and then different profiles. So this allows you to easily type in a gene name and then choose a different profile in order to see which data sets this gene is expressed in. So a profile is just a collection of data sets set to specific criteria. So we have hearing data sets, we have data sets specifically during development of the inner ear, data sets regarding specific single-cell RNA-seq data sets specifically for the zebrafish, for spiral ganglion neurons, so a couple different classes. So in order to search a gene, what you're going to do is type it in your little gene box, and then you click this magnifying glass and choose whichever profile you want. So anytime you're on gEAR, you first walk on, the default hearing profile is what you're going to have. That's just general hearing data sets. So if you go here into the gEAR portal, for example, we're gonna type in SOX. This is the default hearing profile here, and then I'm gonna click search. So what this is gonna do is going to come up with any gene that is related to SOX. So it's taking a little bit longer than it normally does. Sometimes this happens when we have lots of users on all at the same time. But while this is loading, I'll mention one feature. So we have this thing you can see here called exact match. So currently I have this off. So that means that everything that has SOX, any gene name that has SOX in it, is going to come here on this match. I'm not sure why it's not loading. Let me try it one more time. But if you want it to be more specific and only come up with your exact gene, you'll be able to turn this on, which I'll show in a second. Let me see if I can get this loading. If not, we'll just continue to go through the slides in the meantime.
Yeah, you might have to refresh the page. The workshop, people doing the analysis breakout room are really loading the server.
Loading it, yeah. Let's see if I can.
And exact matches come up much faster than non-exact matches.
Okay, I'll show you in the slides of how it comes up with an off exact match, and then I'll actually go in the browser for an exact. So put in my gene name SOX, and then you can see here it comes up with all of these different gene names that had SOX with it, and then all of the displays in your browser. So to go in between these different genes, you just click it, it highlights, and it will come up with all of the data sets and how that gene is expressed. So one important thing to note is the way these data sets are expressed in visualization on gEAR. We have colorized anatomic graphics. So it's displayed from a lighter purple color to a darker purple color, and that's just the intensity of expression. So lighter color means less expression, darker color means higher expression. So you can see here in this beautiful diagram of the cochlea, you have this darker expression is going to be a higher expression in that specific cell type. So for example, the inner border cells here have a higher expression of this gene. So now if you want to look for exact genes specifically, not get that whole list of anything that has that name in it, you'd click this exact match to on. And you can do this for not just looking at one gene at a time, but multiple genes at a time, which is convenient. So if you have, let's say, a long list of genes that you noticed was upregulated in data set, you can copy paste them from your Excel sheet, put them all in, and they'll all search at once. You just need a space bar in between them. So I'll go ahead and try to show you that here now. So I have a list of genes here, separated by a space. And then I want my exact match on. Since I'm on the main page, it comes up as this little target bullseye button. So I click that on, and then I click search. And then it should come up in the search bar again. So since we have so many breakout rooms, so many people using gEAR with especially the analytic tools, it takes a little longer. But on a normal day when there's not a workshop going, it will be very quick. So here you can see in the search results, I have these 4 genes that I put in all listed here. And then to get in between them, I just click. So let's say I want to look at PAL4F3. I click at PAL4F3. And then the data sets change for where PAL4F3 is expressed in these different data sets. So for example, here we have a single cell RNA-seq data set from the Kelly lab. You can see the expression in the hair cells. And then you have many other data sets showing where it's expressed in the hair cells and other locations at various time points and things like that. So this is in just the classic hearing default profile. So another tool, when you're searching, you can use different scopes. They'll yield very different results. So under that exact match little bar, there's also a search bar that you can change the type of scoring method. So this is going to change that coloration you're seeing from the lighter expression to the darker expression. This is going to change how that's scored. So you can see here in this little tool, you press the down arrow, and then you can choose your scoring method based off gene scope, tissue scope, and data set scope. So what that means exactly is you can see here is the gene level scoring is going to be showing you the gene expression based off a specific gene and how it's expressed in specific tissue types for that single gene. Tissue level scoring is going to show your gene expression, how it compares to other genes, but in a specific tissue type. And then data set level scoring is going to show the gene expression of whatever gene you put in compared to all variables in that entire data set. So as I mentioned earlier, one really useful thing about gEAR is you can switch between profiles, so you can really see where your gene is expressed in different data sets that may pertain to you. So in order to switch profiles, you can see at the very top of this left screen here, it says hearing default. Now there's not a little drop-down arrow, but if you click on it, a drop-down arrow will appear. So you just click it, and then you can see multiple different profiles that exist. So for example, if we want to see these genes specifically in development, you click on development, and then it will research the genes that were in your list, and it will come up with those data sets that are in the development profile. So you can see here are these different data sets compared to that hearing default profile. And one other interesting thing is, so out of this list of these profiles you're seeing here, these are all public profiles, but you can also, when you're uploading your own data sets or sharing data sets between other gEAR users, you can also have your own personal profiles yourself. So I have some from my own personal data set here that I can use gEAR to then evaluate. Okay. So another very cool tool is we have linkouts, deafness gene annotation, and context menus. So when you put in a gene, you can actually see here that it comes up with different annotations by organism. So you have mouse, human, zebrafish, chicken, rat, and macaque. So it will come up with the functional annotation here. You get different things like gene symbols. If it goes by any different gene aliases, that will list here. Common Go terms for this. And then in addition to this information, we also have external resource links. So things like UCSC, Unipar Ensemble, PubMed, and then the Big Shield data set. So you can just click on these links. So for example, you want to know more about GFI1 and what comes up in PubMed, you just click PubMed and it will take you right there specifically for GFI1. Rona touched on this before. A very new interesting tool that has recently been added to gEAR is this deafness gene annotation. So if the gene you are searching has any known role in deafness, it will come up with where that known deafness is in either mouse, human, or if there's a mutation that they're currently investigating in human, in this little human with a question mark. So if you here click on that little mouse, it will come up with different phenotypes it has, and then it will take you to MGI to learn more about the different deafness phenotypes and information. I think PAL 4F3 has, here's an example of how it has the different phenotypes listed that have already been found. And it will, for humans, will take you to OMIM and so on. Okay. So within each data set, we have a data set context menu. So in the upper right-hand corner of each data set, there's a little drop-down menu, and then that can give you different displays, views, and so if, specifically if you want more information on that data set, you can, I'm going to just go back to the hearing profile to show this, the auto. So if you want more information on the data set, you're going to click this drop down, and then you can go to info. Sorry, things are taking a bit longer. So it gives you a nice, beautiful, detailed explanation of this data set. So what you're actually looking at it instead of just the visual display. So the abbreviations for all the different cells are listed here, what time point was used. This even has so much detail into exactly how this experiment was performed, what sequencers, and all of that information. This information is provided by the publisher of that data set. But if there's a little bit of information missing, there's something you want more to, you can go to the publication and we'll take you directly to the actual article. Another thing to make it a little bit easier and user-friendly, so the dataset panels are zoomable. So in that same little drop-down menu, you're going to go to the View button on this little magnifying glass with the plus sign, and then that's going to make this dataset full on your screen without any other datasets around, so you can really specifically focus on that one. And then if you want to go back to your main page, you just press the magnifying glass again, and it will bring all of those other data sets up. Okay, another thing to make gEAR a little bit more personalizable is where you're able to switch displays. So you can see here that when you bring up your data set, it has a very specific display of how the gene is expressed in that data set. But let's say you don't want this schematic of the cochlear expression of this gene in the different hair cell types. You want to see it as a violin plot, for example. You're going to click on this display button, and then it will come up as being able to choose. The gEAR has some automatic different displays, as well as you can create your own using the curation tool, which they go about into another section. So you can see here when you click that displays button, the gEAR, as I said, has some of these presets. So here there's a violin plot. So you would just click on the violin plot instead of the cochlear figure here, and then it would change it to that. And then when you see on your main page, it's going to now be this violin plot instead of that cochlear diagram. On the displays, you can constantly change back and forth. So when you change it to one, it's not permanently set that way. You can easily, just within a couple seconds, change it back to the other. So I can show that to you here. So here's your little drop-down menu. You're going to click displays, and then you see this violin plot that gEAR has automatically created. So if you want to switch it to the violin flat instead, click that, close, and then you see it's automatically here. And then you can just as easily switch it right back to the one that was previously there. So when you're creating displays of your own that you may want, there's different options to create scatter plots and multiple other things. Again, that's in one of the other sessions if you're interested. So again, as I keep mentioning, we really want GEAR to be very user-friendly, what you want GEAR to be. So another way to do this is in order to take notes on specific data sets that you can keep track of if you thought of an idea for a data set, didn't want to create another Google Doc or Word doc to take your own notes. You can do this directly on GEAR, and if you're in your logged in account, it will save. So in order to do this, there's a little pencil right next down to the drop menu, and you just click that little pencil. You see, oh, I noticed that the expression happens to be similar throughout this entire data set between these cells for these genes. So you write this in the little notes, type it in, and then you can save it. Now you can save this either privately or publicly. So privately, only you are going to be seeing this note. If you do this publicly, anyone who uses GEAR and has a GEAR account will be able to see that note. So the only time you really want to do something publicly is, let's say it's your own personal data set that you've shared with GEAR, and you want someone to know something specific about that gene expression in your data set, then that would be a good time to make it public. But your own little personal notes, I would make it private for you. And then you just click that save button and it will be stored in GEAR under your own account. A couple of other helpful, useful tools. So we have both a manual that explains everything in GEAR, and you can always get help from our help desk. So for the manual, it's this little book right near your login name. You click on that and there are a whole bunch of different YouTube videos, written manuals for all of the different things that GEAR can do. So something as simple as this basic workshop of using GEAR and the different things it can do, down to uploading your data set, download or being able to analyze single cell RNA-seq data sets of both your own or pre-existing on the GEAR. So there's walkthroughs for basically everything. But if you're still stuck on something, we're here to help. So you can just click on this little bubble next to your name, and then that will bring up this fill out form here where you can type your question or comment. You can also, if you have any suggestions for GEAR, so let's say there's something you're interested in using on GEAR and you notice it's not here, you can type in a suggestion, and it's definitely something we can look into and possibly add, because GEAR is always ever changing and we want to have the tools that everyone needs in this system. So you just fill out your information here, question. If there's any more specific information, you can include that there and submit, and it will create a ticket. So our wonderful GEAR team, including Joshua, will get this and be able to look into it. And with that, if you guys have any questions, please feel free to ask them now. You can show video, unmute, or go in the chat. If there was anything I was confusing, I can go through again, or if you have a question about something I didn't even present, and you're also welcome to ask that. And thank you guys so much for coming today. We're really excited to have so many people interested in using GEAR.
So I have a question how to switch to the room number 2 and when it will be available. So the first session was very informative. Thank you very much for that one. But can we switch to the second one? When will be available?
Yeah, of course. So each session is about 25 minutes. So I think we have a little bit one for this longer, but in the meantime, you can just leave this session and it will take you to the main room, and then they'll tell you when to switch to the next one.
Thank you.
Thank you so much.
Thank you, guys.
Katie, there's one question from Martin
Oh, here, let me pull up the chat.
about if there's any plans to extend the tool to sensory tissues in the future.
I believe we are, yes. Oh wait, is GEAR portal restricted to inner ear tissue, or can we explore expression profiles in the middle and outer ear also? Yeah, I think that's definitely something that Rona has looked into doing. We've also expanded GEAR into its own different database for the brain. It's called NEMO. So I know this is definitely something that we would, any more systems, it's definitely something we can add into. That's something I can talk with Rona about and bring to the GEAR team as well. So I would definitely look out for that in the future.
Yeah, there's certainly nothing about the platform that limits us to, like today, if you had data, you could start uploading it to other tissues. There's nothing preventing that. I think it's just that if we had enough of them, we could create a profile that everybody sees on the front page once there's enough curated data sets in another tissue.
Yeah, but if you, like Joshua was just saying, if you have your own data set that pertains to middle or outer ear, before we get profiles from other people, you're welcome to upload that either privately on your own account or publicly. I've had friends that work a lot in neuroscience, so they do a lot with just brain. And even before NEMO was created, they were using the GEAR to analyze their own RNA-seq data sets, just because the tools are so applicable to any data set. With scoring method demo, you pulled up tables on screen with values instead of the graphical representation. How did you do that? Joshua, would you be able to explain this a little bit more? You're a little bit more familiar with it than I am.
Yeah, and I apologize for my voice. It's going away for some reason. So in that demo slide, I had just created screenshots of what the original source data looked like before it was uploaded to the GEAR. There's not a table view so clearly like that you could launch. But for most of the displays, like the SVG displays or anything like that, if you hover over any particular part of it, you'll see the actual values there. So that's one way you can see the values directly. We will be adding soon the ability to download all the source data, but we want authors to be able to choose whether that's okay for them to do or not. So I'm adding that feature where when people upload, they say whether the full data sets can also be downloaded. And then the next one, how to cite GEAR data in the paper or GEAR the application in a paper.
So anytime you use GEAR, we use the umgear.org and then retrieved by date. And then if you're specifically looking, you've retrieved data from a specific, any data set you use, you also cite that original publication. So for example, if you're using GEAR and then you were looking at the expression of a gene and you happen to use the Matt Kelly data set for that, then you would also cite that publication from Matt Kelly's lab as well. And if you have more questions on it, let me know. I can give you examples because I've had to cite it in a couple of my papers as well.
Yeah, and the GEAR itself actually has publication pending. So we'll soon have a link at the bottom of the page for the GEAR itself.
Will you also add the inner ear gene pathway analysis as a function? That is something I know we've talked about before. I'm not sure. I don't think it's in the works currently.
Yeah, that's not an active thing. I think it's a good idea. It's not an active one.
It's going to take a bit more work of power to be able to do that. But it's something we use separately in our own browsers, so definitely something we're interested in. Yeah, Daisy, do you have an email? If you put it in the chat, I can email you an example. I'll pull it up from mine. Thank you. No problem. Is there anyone who's, oh, is it okay to switch to breakout 3 without knowledge of breakout 2? Yes. They're both different analysis tools, but they can be done separate from each other. So it doesn't matter if you know how to do one versus the other. No problem.